Esteemed guest speaker for this scientific session, Dr. Ashok Kumar Janakiraman, Faculty of Pharmaceutical Sciences at UCSI University, Malaysia. Dear participants, ladies and gentlemen, good afternoon. I, Dr. Shagufta Khan, on behalf of the Institute of Pharmaceutical Education and Research, welcome you all to the last scientific session of the 2 days APTI Maharashtra State sponsored National Seminar on Nanotechnology and Healthcare. I believe with confidence that every one of you has made the most of this platform to enrich your knowledge of considerable research on nanotechnology in drug delivery and diagnosis. There will soon be another brainstorming session for you, for which we are honored to have a great researcher and an academician, Dr. Janakiraman. I express my gratitude to you, sir, for accepting our invitation and spending time for enriching us. I am indeed elated to introduce our guest to the participants. Dr. Ashok Kumar Janakiraman is an assistant professor in the Faculty of Pharmaceutical Sciences at UCSI University, Malaysia, which is ranked 284 in the QS World University Rankings for 2023. Ashok received the MPharm Pharmaceutics and PhD Pharmaceutics degrees from Sastra and Prist University, Tanjavur, India in 2008 and 2014, respectively. His research is situated in the field of pharmaceutical technology and product development with a special focus on nanotechnology for drug solubility enhancement, fast melting tablets, topical products, and collaborative R&D in industries such as pharmaceuticals and cosmeceuticals. Dr. Ashok teaches several courses on pharmaceutical technology to bachelor's and master's pharmacy students. He is the author or co-author of more than 50 papers in international refereed journals and more than 25 conference contributions, 4 patents, 1 trademark, 2 book chapters. He has received several important recognitions for his research career, including the ASEAN Youth Innovation Award in Malaysia Technology Expo 2020, the gold medal in the PECI-PTA 2022 International Conference, and the exposition on inventions by institutions of higher learning in Malaysia. He received the UCSI University's Research Excellence Award 2022 in the Top Research Publication Contribution category. He currently serves as the Technical Advisor Academic for Geomatica University College, a Fellow of the Einstein Research Academy, and an Associate Member of the Malaysian Pharmaceutical Society. He secured a 3-university internal grant worth Malaysian ringgit 1,24,000, which amounts to about 20 lakhs in Indian currency from the Ministry of Higher Education Malaysia, a fundamental research grant scheme worth ringgit Malaysia 89,000 that amounts to about 15 lakhs in Indian currency, an industry consultancy project worth ringgit Malaysia 2,40,000 that amounts to about more than 40 lakhs in Indian currency. He currently serves as Vice Chair, Postgraduate Research cum Faculty Research Scholarly Activity. So dear participants, join me welcoming Dr. Janakiraman. Dr. Janakiraman to talk today on metallopolymer nanoparticles in cancer therapy. So may I request Dr. Ashok Kumar Janakiraman to start with his presentation. Dr. Janki Raman, are you able to hear me?
Sorry, madam, I was muted. So now, can you hear me, right?
Yeah.
Okay, the slides also visible, right?
Yes, yes.
Okay. Thank you, ma'am. Thank you, Sankuta Khan, ma'am, for your warm welcome and the introduction. I would like to thank the management of Institute of Pharmaceutical Education and Research and Professor Rajendra Gashwal, principal of this institution, to giving me the opportunity to share the views on metallo nanoparticle for the cancer therapy. So I'm very proud to be a part of this conference. Actually, I am also one of the member of APTI. My registration number is 432. I was a member from past 13 years from this APTA. So I'm very happy to be a part of this conference. So my voice is audible, right? Shall I continue? Hello. Hello.
What, sir?
Yeah, shall I continue?
Yes.
Oh, okay. Thank you, ma'am.
Sir, sorry to interrupt you. I think your video is closed. Sir.
Is it 1 minute?
Switch on your video, sir.
Yeah. So later, after a few minutes, maybe the background will get. So thank you for your warm welcome and introduction, ma'am. I think I can be able to start my lecture topic on metallopolymer nanoparticles for the cancer therapy. So
Yes, sir.
I hope. Sorry, ma'am.
Yes, sir.
Okay.
You can start with your speech. Yeah.
Okay, thank you. So I hope everyone in the past, yesterday, they will talk about the cancer, how it may impact the nanoparticles or nanotechnology is impact on the healthcare system. Mine is totally is based on the drug delivery, how the nanoparticle impact on the drug delivery to treat the cancers. Sorry, madam. Okay. So in my lecture topic, maybe I'm going to cover the prevalence of cancer. What is the current situation? How many people are going to affected or getting affected by the cancer? And what are the current treatments available for the cancer treatment? And what are the drawbacks associated with the cancer treatment? And maybe I am focusing here on the metallopolymer nanoparticle. Actually, this is a new concept. Nowadays, they are focusing to make a target therapy, especially for the cancer treatment. And in that, you should know, you are going to know about what is made by metallopolymeric nanoparticle, what are the benefits and drawbacks, and how to choose the polymer and metals while you prepare as a nanoparticle, and what are the strategies in introducing the metal to the polymers, and what are the types of synthesis, that means the method to prepare the nanoparticles, and what is the application, and how many of the products is already available in the market. Maybe we are going to cover the overview of the metallopolymer nanoparticles. So firstly, what is the prevalence of the cancer? So recently, Globocon 2020, that means the global cancer burden, the report from the International Agency of Research on Cancer, they are saying that the worldwide more than 20 million cases are discovered annually around the world. It's, you think about how many people are going to get affected for the cancer. It's around 20 million. At the same time, the death was observed around 10 million people every year are dying due to the cancers. So if you look at this graph, we have the different types of cancers. It's like the topmost prevalence in the cancer is a breast cancer, then followed by lung cancer, colorectal, and prostate and the stomach cancers. As per the WHO 2019, they are saying that the cancer is the first or second leading cause of death before the age of 70. So as per the WHO data, is a maximum people, the majority of people affected from the African country, followed by the Central America, Eastern Asia, and South Asia, South Central Asia. So we are in the fourth position. That means in the fourth leading region is affected by the cancers. So as you know, we are treating with the cancer in a numerous cancer treatments available currently. We will say that the chemotherapy, we have a lot of drugs and the natural plant sources we are using to treat the chemotherapy, and also we have hormonal therapies, immunotherapies, and bone marrow transplantation. If it is in the third or fourth stages, they will prefer to go with the bone marrow transplantation. And we are nowadays we are focusing on the targeted therapy. So our nanoparticle also is a part of targeted therapy to treat the cancers. And we have radiotherapies and surgeries. In the worst case, we will choose the surgeries to remove the organs, the cancer affected area. So these are all the current treatment options we are doing or we are using for the cancer treatment. So what are the limitations with the current treatment? Here I'm going to focus on the delivery system. Is most of the treatments available in the different dosage form is unable to achieve the cancer-free status. Even though if you have some multiple functional treatment, we cannot achieve the cancer-free status. Still, the day by day the cases are increasing, and the prevalence of a cancer is increasing. As I shown in the previous slide, also is a 20 million people per year annually. So it's a huge number. It's not a small. Then the treatment currently available treatment it's showing is low tumor selectivity. So what, even though if you have a nanoparticles or different kind of drug delivery, conventional drug deliveries, we cannot be able to choose the cancer cells, meaning that we are missing here as a targeting of tissues or organs, cancerous tissues or organs. And of course, this is a multi-drug resistant cell, same like antibacterial resistant. Here also the drug which is already taken for the cancer. So after usage of few years, that make that organ or cells make a resistant to particular drug. So we need some special kind of effect to overcome the drug resistance in the cells or organs, especially for the antibacterial and cancer. And of course, the drug which is having the pharmacokinetic and biodistribution and rapid metabolism and excretion from our body. Even though if you're taking a drug into the different dosage form, is like oral, or if it is is a intravenous or extravascular administration, due to its poor pharmacokinetic and distribution and rapid metabolism and excretion, it can be easily removed from our body. As you know, as a pharmacy student, you know what is meant by pharmacokinetic, right? ADME, absorption, distribution, metabolism, and excretion. So this is especially for the cancer drug, we cannot control these things. It can be easily eliminated from our body. Then the another limitation here is off target toxicity. Suppose if you are taking a drug, we cannot get 100% of drug into the targeted places. Mostly half of its quantity will be eliminated through metabolism or other excretion functions. So we are getting the drug in a half quantity into the target place, so that half a quantity will not be enough active ingredient or enough drug to cure the cancers. Then of course, some of the drugs, not some, most of the drugs having the adverse effect, so it may produce us the side effect on the healthy organism. Suppose we are taking the drug for cancerous or affected organs, even though that drug will affect, that means suppose if you are taking any drug, that also will affect the healthy organs or healthy cells, and sometimes it may produce fetal death also. So based on those limitation, so we need to come up with some targeted or suitable drug delivery system to address those limitations. So as I said, the targeted treatment for the cancer or affected cells is reduces doses needed now. So we can choose the nanoparticle as one of the solution to overcome the limitation which we have with the current treatment for a cancer. So in that nanoparticles, we have different types of nanoparticles. This is the first category is a polymer based nanoparticles. This polymer based means it's having the polymeric molecules. Their molecular weight is maybe few thousand Daltons. So we have a dendrimers. Dendrimers is nothing but a polymeric molecule. It also used to treat the cancer. Then we have a polymeric measles. Measles is nothing but a group of molecules. So the chain of molecules. Then we have protein nanoparticles, nanospheres. So these are all comes under the polymer based nanoparticle to treat the cancers. Then the second category we can say is based on the it's an inorganic inorganic metal type nanoparticles. In that metal we have gold, silver, platinum, and copper. So some of the metals I mentioned here, but we are using a lot of metals to cure or to treat the cancers. Sometimes we will use the metal oxide, is zinc oxide or titanium dioxide. So in that, the zinc oxide is my area. I'm currently doing the research work on that zinc oxide to treat the cancer. Mostly our work is on in vitro stage. We are using the cell line studies, but that's also is considered as an in vitro characterization to prove the cancer activity of my zinc oxide nanoparticles. Then we have silica, that is also a inorganic material used for cancer treatment. Then the other category is a lipid based. So here is a liposomes, solid lipid nanoparticles, lipoflex. This and all is example for a nanoparticle, lipid based nanoparticles. So that also can be used to treat the nanoparticle. We can make them or we can convert them into the nanoparticles in a easy way, and it can be treat the cancers. Then the fourth category is a carbon based, that is fluorine, carbon nanotubes, and graphenes. These are all is a carbon molecule is used to treat the nanoparticles. So out of this, the organic metal and metal oxide is called as transition metal elements. So we have a lot of metal elements, but specifically the metal element which is having the inorganic characteristic is called as the transitional metal element. So transitional, we can say that it will move from d orbit to s or s to p. It's like the electron donation will be higher and it can be moved from one orbit to other orbit. That's why we can call it as this is a transitional transitional metals. So the nanoparticle, as you know, the nanoparticles is having the unique surface characteristic, right? So unique surface characteristic, and also the particle size is in a nanoscale range. So this is maybe we can consider as this is the advantage or disadvantages also. Maybe we can say due to its surface characteristic, the nanoparticles size both will make them into a limitation or is a ineffective treatment for the cancers. So what we need to do? So we need to do the improvement, or we have to do some stabilizing agents like polymer, surfactant, or polysaccharides we can include along with the normal nanoparticle to enhance its functional characteristic. But I trying to say here, even if it is in the nanoparticle, it may not be able to make or treat the cancer due to its surface characteristic, meaning that if the surface characteristic, it can be easily captured by the reticuloendothelial system. It's like in our organs like spleen, liver. So that can be easily removed from our body. So if you administer in the form of nanoparticle, it cannot be stay longer time in our body to act against the cancer. So it can be easily removed from our body through the reticuloendothelial system mechanism. So nowadays the researchers are focusing how we can able to make the nanoparticle or make them nanoparticles to stay in our body for a long duration to prevent such kind of elimination from our body or excretion from our body. So in that, this is one of the newest strategy is the metallopolymer nanoparticle. So here we can use polymer to stabilize the nanoparticle, and also the size will be bigger as compared to the normal nanoparticle. So if the size is bigger, maybe it will be stay for a longer time in our body, and it may produce the required pharmacological response, or maybe it can be easily treat, or it can reach us the target of cancer cells. So we have nanocomposite, the metallopolymer nanocomposite. This is combining the properties of metal and polymer, so we can have a synergistic effect. So some of the metal particles, for example zinc oxide itself having an anti-cancer activity. Suppose if you give this metal oxide with some polymer, so it can be like a matrix kind of nanoparticles, so it can be go and target the cancerous cells or required place to produce a desired pharmacological response. So if you add other drugs also, anti-cancer drug in along with the metal and polymer, that may produce a synergistic effect with the target activity. So for your information, so each metals and polymers, they have their own advantages and disadvantages. Let's say if the metal, if you are using different metal, for example gold, silver, zinc, copper, aluminum, or titanium, they have their own advantages and disadvantages. This is just I am mentioning here as a common advantages and disadvantages of the metals and polymers. So if you look at the metals, it's a unique optical, electrical, and magnetical properties. So this is an advantage. Due to this electrical and magnetic property, we can make it in a nanoparticles, and variety of potential choice, like it having a different groups of metals to transition metals and lanthanide metal. Just now I told what is meant by transitional metal. Mostly what we are using here for a metallopolymeric nanoparticle is a transitional kind of metals. And the advantages with the metal oxide, it may produce or enhances the radical oxygen species generation. So this is one of the main mechanism to kill the cancer cell. We can call it as ROS, reactive oxygen species generation. So it may enhances the radical oxygen species generation. So through this, most of the cancerous cells will be removed or will be killed. Some disadvantages with the metal nanoparticles, it may be produce a toxicity. If it depends on the particle size. If it is a very small particle size, definitely it may produce a cell toxicity. We can call it as there is a nanotoxicity. And also the plain metal oxide will produce off-target interaction. So it may not be deliver 100% of drug through this metal oxide. It may produce off-target interaction and lack colloidal stability. So normally So we are working a lot of, we are doing with the research, we are doing a lot of nanoparticle research, but very few of the molecules only come to the market due to its stability. So main drawback with the metal nanoparticles is a stability issue. Uh, then we move to the polymers. So the polymers having a lot of advantages. So it is a diversity of choice, natural and synthetic polymers we have. So it's having the good surface functionalization, meaning that we can add any kind of ligands attached with these polymers to target the cancerous cells. And it also improve the physical properties of the metal. Suppose if you give polymer along with the metal, it may improve the physical properties and also increases the stability and solubility and stability of metals. It reduces the cytotoxicity associated with the metal. As I said, it's having the toxic effect, right? Suppose if you give along with the polymer, we can overcome the cytotoxicity associated with the metal, especially for a nanotoxicity or the normal cell cytotoxicity can be overcome by using the metal with polymers. And it having the payload flexibility. What it means? Payload flexibility means we can load the high amount of or high quantity of drug into the polymer matrix. So we can say that this is a payload flexibility. If you want small quantity, we can reduce the quantity, and also we can increase or enhance the drug availability in the polymer matrix. Of course, the polymer with metal is lower cost as compared to go with the metal alone. The disadvantages with the polymers is a possibility of aggregation, and sometimes it may also produce some toxicity to the normal skins or normal organs. Okay, I think everyone know, right? What is this? I hope everyone know this table. We will call it as this is a periodic table, right? Yeah, periodic table is actually having metals, metalloids, and non-metals. Sometimes some gases also, noble gases also, halogen. So everything is there in the periodic table. So if you look at that area, the middle area and the left side corner of this periodic table is almost all the blue color or all are metals. It starts from lithium until the LR. Okay, so we have different metals, metalloids, non-metals, halogen, and the noble gases in the periodic tables. So if you look at this table, it's around 75% of the elements are the metals. The remaining may be the metalloids or gases. So almost 75% of element in the periodic tables are metals. So some of the metals like gold, silver, or zinc and copper, it can be used for the metallopolymeric nanoparticles. Even though if you have 75% of element in the periodic table, sorry, metals in the periodic table, so we are using few because of its characteristic, and it is more suitable to make in a form of nanoparticles. So some of the polymers, as I said previously, we have synthetic and natural polymers. These are all the example: polyesters, polypeptides, recombinant protein-based polymers. We are using. These are all the synthetic polymer to make a metallopolymeric nanoparticles. Then we have some natural polymers as well, like proteins, peptides, and polysaccharides. So these are all natural, so it can be easily biocompatible it's a no issue for the digestion. So it can be easily get digest with our normal biological function. So no worry about the elimination issue or deposition. So it can be easily degraded in our biological system. The synthetic polymers is have a big batch size synthesis. We have a different long chain synthetic polymers, and is insignificant batch to batch variation. So suppose it don't have much variation if you prepare in a different batches. The disadvantages of the synthetic polymers, all type of polymers have the some level of heterogeneity. It's like a toxicity. It's difficult in the synthesis and characterization for a synthetic polymers. The natural polymers is of course it's improve the pharmacokinetic, increase the biodistribution of a therapeutic agent. It's avoid hazardous organic solvents because it's a natural one. It get easily biocompatible with our biological system. The disadvantages for this natural polymers, it's a potential microbial contamination due to the natural sources. Of course, if you are derived from the natural source, it's possible to get the microbial attack. And sometimes the natural available polymers should have a heavy metal contamination that also will make or produce a toxicity. Then here also we can face the batch to batch variation. Suppose if you are preparing many batches, each batch is having a different characteristic or loading capacity. Impurities leading to the immune reaction because it's from the natural source, so possible to get impurities that may cause immune reaction in our body. As you know, if it is any foreign particle enter into the body, so body will immediately react, right? So is also is considered as impurity that make a immune response. So now we move to the strategies. What are the strategies to prepare the metallopolymeric nanoparticles? So we have a three concept here. So one is a surface coating of metallic surface coating of a metallic core by the physical linkage or layer by layer. Okay, so we will have three different strategy here. First one is a surface coating of metallic core by the physical linkage or layer by layer. You look at that picture, it's the type 1 picture. It's attached with the physical linkages. So the linkages may be the polymer. So sorry, this is a polymeric backbone is considered, the green color is considered as a polymeric backbone. It's attached with the metal ions or metal elements. So through the linkages, the physical linkages, maybe we can use some ligand or some kind of polymers to attach with the metal ions. So the first type is an example for surface coating of a metallic core by physical linkage, or maybe we can have a different layer by layer mechanism to to modify the surface coating or to make a surface coating with the metal ions. Then the second one is a entrapment within polymeric matrix. So you see the second picture type 2, the green color is a polymer backbone. So just this one is attached with the metal center. So the yellow color is a metal ions or metal elements. So it's simply stick on the polymers. Then third one is considered as a utilization of metals on the polymeric capsule. So if you look at the third picture, it's like the green color, then the yellow color metal, green color polymer, again the yellow color metal. So it's like a capsulation or encapsulation. So it's almost like a mixture of 1:1 ratio, like metal with polymers. Okay, now you should know, right? But what are the strategies to create or making the metallic nanoparticles? So these are all the different synthesis approaches. Synthesis means how we are making the nanoparticles. So these are all the different methods or approaches to make the polymeric nanoparticles. The first one, maybe we can say this is a solution to gel, meaning that the simple solution we will convert into the gel matrix or cohesive gels by using the polymer and metal ions or metals inside. So the solution will be converted. Normally we will add the polymer. We can convert the solution into the cohesive gel-like structure. So this first mechanism, the synthesis mechanism or approach is a salt to gel conversion. Then the second one is a reduction. So reduction is nothing but the chemical atom into the non-polar media. We will convert or we will reduce them by using the stabilizing agent. The stabilizing agent may be here used as a metal ion. So we are the chemical atom in the non-polar media will be reduced by using the chemical reaction or using the stabilizing agent. So the advantages with this reduction method is actually we can prepare the nanoparticle in the controlled size. For example, if you want 50 to 100 nanometer to achieve the target place or target region, so we can prepare the nanoparticle with the same size. So we can prepare in a controlled size, and of course this method is more cost effective than the other method. It's a simple reduction by using the stabilizing agent. Example for the reduction method is a dendrimers. So it's an example is a dendrimer for a reduction method. So next one is a co-precipitation method. So co-precipitation method, here we can add 2 or 3 cations in a solution. Then it form a precipitate. Okay, if you mix 2 or 3 cations in a solution, it produce directly, it precipitate the ions, metal ions. So here in this case we can add 2 or 3 metals also. So it's like a combination, more than 1 or 2 cations in the solution, then it may be directly get precipitated. There is a co-precipitation method. So next one is a template synthesis. Is like a self-assembly mono layers. Is normally it's a polymeric matrix. You look at that, we have a cavity on the template synthesis. The drug can be loaded onto the cavity. It's a self-assembled monolayer metal nanoparticle within the polymeric matrix. So you see the blue color one is a polymeric materials. It can be the drug or metal ion can be incorporated into the polymer matrix. Then this one, the next one is a layer by layer assembly. So layer by layer assembly means here is actually sequentially depositing the oppositively charged species. For example, positive, negative, positive, like that we can sequentially assigning the species. Then it's like a sensor. It's normally it's used as a sensor to identify the cancer environment. Okay, this is called as a layer by layer assembly. Then the next one is a well-known polymerization technique. So polymerization is like a simultaneous polymerization of monomer and reduction of metal ion. So if you add monomer with the metal ion, it may produce reduction. So finally, it will form like a metal nanoparticles. The polymerization more common method used for a polymerization is emulsion method. Okay, the next method is electrospinning. This is like a electrical field is applied to the polymer solution. It may converted the polymer solution will converted to the nanofibers. Okay, electrospinning, we will just apply the electric field to the polymeric solution. Then that polymeric solution will convert into the nanofibers. Then gamma radiation, radiation, you know, right? It create the if you pass any radiation or light into the solution, it create a reducing or acting agent in the solution. Then it may be converted into the nanoparticles. We have an example later. We are going to discuss about this gamma radiation for a photo lighting cancer therapy also. So these are all the general synthesis approach for the metallopolymeric nanoparticles. Is there any questions or doubts so far? So far, what we discussed here is what is meant by metallo nanoparticles. What are the metals we are using to prepare the nanoparticles, and examples of natural and synthetic polymers and metals. Then what are the synthesis approach to make a metallo nanoparticle, polymeric nanoparticles. Then this is the important part of my today's presentation. So it may be applicable. That is application how the nanomaterial is used in the healthcare system. Application of metallopolymeric nanoparticle in the cancer treatment. So we can use this metallopolymeric nanoparticle in drug delivery, therapeutic drugs. Sometimes the metal nanoparticle itself it's act as a drug, and also it's used as a drug delivery tool. Maybe it's a nano carrier to bring the drug from the administration site until the targeted site. Then it may be act as both drug delivery as well as the therapeutic drugs. And biosensor. Biosensor is like it's like a diagnostic tool. So firstly, we will identify the cancer in a early stage. It's a kind of diagnosis method. So it may be used as a biosensor and also the theranostic agent. What is meant by theranostic? Theranostic is nothing but the concept of diagnostic and treatment by using the one one concept. So diagnosis and treatment will be do it by one concept, that is called as a theranostic. So this metallopolymeric nanoparticle also used to do the is also act as a theranostic agent. And this nanoparticles may be useful in a additive therapy. That means kind of additional or synergistic therapy, and also it's used in a bioimaging, meaning that they can take a photo or imaging of a affected area to diagnosis or to find the find the nature of cancer, or we can easily identify the stage of cancer by using the bioimaging. Then in the upcoming slides, we are going to have a detailed discussion on each application. Yeah, I hope I answered for your question. What is meant by theranostic? When the agent which is having both diagnostic and therapeutic action is called as the theranostic. Okay, I hope answered for your question. So the first application is a nano carrier. So the first example given here is a red light responsive metallopolymer nanocarriers with conjugated with the encapsulated drug. So this is example for the phototherapy against the multi-drug resistant tumor. As you know, right, what is meant by multi-drug resistant tumor? Because it's already available conventional treatment, we are using some medication, right? Chemotherapy. So after using the same chemotherapy drug for a longer duration, your cancer cell will not react for or will not will not respond for the drug. So that is called as a multi-drug resistant tumor. Same way, if you change the drug also after certain period, it can be considered as a resistant. It won't react for that drug. So in this case, we can use like a phototherapy. Uh, Here, the polymer, the same concept used the metallopolymeric nanoparticle concept we are using to treat the multi-drug resistant tumor. So the polymer used here is a PEG, polyethylene glycol. The metal used as a ruthenium. Ruthenium also is a transitional element or transitional metal. The drug is a chloramfenicil and doxorubicin is a well-known anti-cancer drugs. So the red light, how we can use this one as a nano carrier. So the red light radiation trigger the release of chloramfenicil conjugated ruthenium moieties and release the doxorubicin in the MC7 cancer cell line. So when we administered the drug into the in vitro cell line, the red light radiation trigger, if you make a light on that, it may trigger the release of drug from the metal moiety into the cancer cell line. So the doxorubicin encapsulated polyrubin chloramphenicol missiles demonstrate enhance the intracellular uptake. So normally, because this is already is a resistant tumor cell, so it cannot be respond for chloramfenicil or doxorubicin separately. But if you give it in the form of metal polymeric nanoparticle, then it can be activated. That means it may produce a synergistic effect on the growth of multi-resistant cancer cell. So meaning that it can be kill the cancerous cell by different mechanism other than the normal doxorubicin or chlorambucil mechanism. It may be easily move into the cancer cell because of its polymeric and metal ruthenium characteristic. Then it reaches the resistant tumor cell and release the drug. So in this case, we can kill or prevent the cancerous cell. So this mechanism is given here in a illustration. So if it is give it the metal ion, there is a poly RuCHL, that is a polymeric ruthenium drug complex. We will prepare the first step, how we are preparing the polymeric metal nanoparticle. The second picture, B picture, is showing how after the red light radiation, how the drug release will be there from a nano carrier to target the cancer cells. So I given the cite the authors, those who published this paper. Maybe if you want a more information, then you can go and get further information from the published paper. The sequential dual modal strategy based on the photoactivated metallopolymer for the on-demand release of photosensitizer and anti-cancer drug. This is a different concept. The previous one is a polyethylene glycol, right? So this one is a methoxy polyethylene glycol and piperidine functionalized polycarbonate. So PTM CP, we can call it, but we are using the same metal, ruthenium. They use the same metal, right? Ruthenium, and the drug is different. It's not a previous one. This is a different drug. Paclitaxel. Paclitaxel also, yeah, anti-cancer drug. So the polymer self-assembled into the nanoparticle and accumulated at the tumor site. So you look at the picture, the same picture. Here we elaborated how it can be reaches into the tumor cell. Okay, the polymer self-assembles into the nanoparticles. It accumulated at the tumor site. Red light radiation at the tumor directly cleave the drug. That means the drug on the ruthenium complex will release the drug, and also it produce the oxidation O2 for the photodynamic therapy. So the O2, the generated O2 trigger the breakage of linkers to release the drug, paclitaxel drug for the chemotherapy. So this is the mechanism behind that, how the drug can be cleaved. That means how can be released from the metallopolymeric nanoparticles. Okay, when the red light radiation at the tumor directly cleave the RU complex and produce the oxygen for the photodynamic therapy. So generate the oxygen triggers the breakage of linkage to release the drug. That is paclitaxel for the chemotherapy. So cell viability of cancer cells treated with the ruthenium nanoparticles and red light was 32.4%, which was 1.8 folder lower than the cell in a ruthenium and nanoparticles and red light. So meaning that if you added the drug, then it may be the higher fold of that means the cell viability in the cancer cell treated with the complex in a red light was 32.4%. It is lower than the cell with the plain complex polymer metal complex. So the drug will play an important role to release or to prevent the cell viability. I hope you understood what I'm trying to say here. The mechanism for the methoxy polyethylene glycol and piperidine functionalized polycarbonate ruthenium paclitaxel complex or nanocarrier. So the second one is actually is a therapeutic. So this is fighting against the drug-resistant tumor using the dual responsive PT4 and ruthenium-2 bimetallic polymers by Zheng et al. So the author used here as a two metallic polymers to target the drug-resistant tumor. The polymer used here is a polyethylene glycol, and the metal used here is a platinum-4 and ruthenium. So the previous one they used a single polymer, single metal and single polymer. Now they are using dual metal. So that is a bimetallic polymers. So from this therapeutic concept, the irradiation of the cancer cell consisting of poly platinum and ruthenium nanoparticles with the red light generate oxygen. The concept was the photo generative concept. It generates the oxygen again. It's induced the polymer degradation and trigger the release of ruthenium anti-cancer agent So the anti-cancer agent used in this concept is a cisplatin. It's released in the intracellular environment by reduction of a platinum 4 moieties. So lower increase in tumor volume with this complex, the polyplatinum ruthenium drug complex, as compared to the cisplatin treatment in the PDX tumor breathing mice. So as compared to that, so lower increase in the tumor volume when we go with the drug complex. So polymer metallic drug complex. Then it is having the good bioavailability. So if you look at the picture, there is actually is a tumor volume. So if you, this is the black color is indicate that this is a normal tumor volume. So if you give it in the form of plain drug, or we can use only with the polymer metallic compound. The polymer metallic compound with light should have a more effect. So you see that the dark green color, that is actually is a volume, tumor volume. So the tumor volume was suppressed when we are treating the polymer metal oxide with drug plus light. So it's mostly the photo light concept with the polymetallic polymer metallic nanoparticles. So the same thing they are give it in the form of graph. So that also you can easily understand that the poly platinum ruthenium complex plus light, it's showing the maximum effect. That means it increases the tumor volume. So it's lower the increased tumor volume. So next therapeutic agent concept we will use here as a bio-inspired synthesis of chitosan with the copper oxide nanoparticle using routine and their anti-proliferatory activity in the human lungs cancer cell lines. The polymer used is chitosan. The metal is a copper oxide. So we can use the synthesized copper oxide with the drug, routine drug. So its non-composite showed the concentration depends on anti-proliferating activity against F549 cancer cells, and their IC50 value was found to be 20 plus or minus 0.5 microgram per ml. So if you look at the graph, there's a routine is an alone, it won't produce much IC50. So maybe it's the IC50 value is around 55 or something. That means we need a higher dose to cure the cancer. But if you go with the complex, so the complex with chitosan and copper oxide, the value given here is the IC50 value is given here is at 20 only. So it's almost 3 times lower than the routine alone. So this is a mechanism how we can prepare the metallopolymeric nanoparticle, chitosan copper oxide metallocopper oxide nanoparticles. So each concept I given the author's details. So if you want further information or elaborate information, then maybe you can refer those cited papers. So next therapeutic agent here is a self-assembly of copper polypyrrolidine supramolecule metallo-nanoparticles to achieve the enhanced anti-cancer efficacy. Here again, we will use different polymer, 2-phenyl imidazole, phenylethylamine PIP, and the metal used here is a copper metal. What we observed from this concept is successfully developed a simple copper-2 complex is capable of self-assembling into the metallo-nanoparticle. It's like a layer by layer concept. It's having the self-assembled into a metallopolymeric concept. You remember in the synthesis method I told you, right? It's like self-assembled synthesis. So this is prepared based on that concept. The complex enters into the cancer cell via endocytosis. So endocytosis, you know, right, engulfing. It's effectively inhibit the cancer cell with no significant toxicity effect on the normal cell. So we achieved, maybe here is what we really needed. So it should not be affect the normal cell. So from this concept, the complex enters into the cancer cell via endocytosis. It effectively inhibit the cancer cell alone. So it won't affect or it won't produce any significant toxicity effect on the normal cell, which we wanted actually in the needed therapy, and demonstrate the dose and time dependent inhibition of the tumor growth. So if you look at the graph here, so it depends on the concentration. If you increase the concentration, we can get the more anti-cancer activity. So normally they will use the rodents to check the anti-cancer activity. Sometimes they will use the cancer cell lines, available cancer cell line, to check against its efficacy. The next concept is a pH responsive chitosan acrylamide gold nanocomposite. Is supported by with a silver nanoparticle for the controlled release of anti-cancer drugs. The polymer used here is a chitosan graft with the acrylamide monomers. The metal combination of metal again, gold and silver. Drug used here is a fluorouracil, 5-fluorouracil. The observation from this concept, it successfully prepared pH responsive bimetallic nanocomposite hydrogel via the gamma radiation method. So meaning that if prepared things will respond only in a certain pH range. So the presence of gold nanoparticles reduces the cytotoxicity of silver nanoparticle while increasing the ability to kill the high number of liver cancer cells. So gold silver nanoparticle increase the releasing efficacy when CSG, PAM and silver gold with silver nanoparticle loaded with a fluorouracil anti-cancer drug. Actually, if you look at the graph here, this maybe have 3 different concept. In that, the drug loaded metal polymeric nanoparticles showing the higher efficacy. So this is how they prepare the complex. That means the metal polymeric nano composite. They given in a brief manner for a easy understanding. It's in the illustration picture. The third concept is a nano carrier plus therapeutic agent. So the polymer coated gold nano cells for the combinational phytochemistry, phytohemotherapy of a pancreatic cancer with the gemcitabine is from the Emamash et al. Is the author. He proposed this nano carrier plus therapeutic agent. So the polymeric metallic nanoparticles is act as a nano carrier as well as to treat the cancer. So therapeutic both. So polymer used here is polyethylene glycol and polymethyl ether thiol. And the metal is gold. They also included the gemcitabine as a drug to treat the cancer, pancreatic cancer. The observation from this study, it's demonstrated the effect of different polymer chain length on the cellular uptake of gold. combinational effect of the drug and the local hyperthermia by gold as induced via laser. This results in highly toxic potency in a MAIA-PaCa2 cell, which was considerably higher than the parent drug and also had a longer lasting effect. So if you make it in the form of polymeric nanoparticles, then it may be produce a longer duration effect. So they are comparing their effect with the nanoparticle treatment group, then nanoparticle with the laser, plain laser or drug with laser treatment. So in that they found that the drug with polymeric nanoparticle plus laser is having the highest inhibition, cancer cell inhibition. The fourth one is a biosensor. As I told you, this is actually is a detect the diseases in the early stage. So the polymer used is a polystyrene. Ruthenium is a metal used to prepare the metallo polymeric with the different ligands for biosensor action. So synthesis 3 metallopolymeric nanoparticle branch with the luminescent oxygen probe like RU1, RU2, and RU3 by adjusting the pi conjugation length of ligand. So we have a different ligand length with the three metallic polymeric branched nanoparticles. So in that RU3 nanoparticle exhibit the highest oxygen sensitivity, 82%, as a functioning as a luminescent sensor. So out of these three, the R3 one is showing the highest oxygen sensitivity. So able to image the hypoxia within the multicellular tumor spheroid. So that can be used to find the initial stage, or even if it is within the multicellular, also we can find the nature and also the status of a cancer, and also we can find the stage, which stage of cancer is there in the cells. So this is mainly considered as a diagnostic tool. Biosensor is a diagnostic tool to found the cancers in early stage. Then the theranostic part, as I told you, the theranostic means it's having both diagnostic as well as the treatment option. So polyethylene glycolate, gold, platinum, nanodendrimers as a novel theranostic agent for the computer tomography imaging and photothermal radiation synergistic therapy. The polymer used in this study is a polyethylene glycol. They used two different metals, gold and platinum. So gold, platinum, nanodendrimers exhibit no acute cytotoxicity to the both HUVEC cells and 4T1 cells at the test concentration. That means in the 75 microgram per ml, it won't produce any cytotoxicity, no acute cytotoxicity on the cell lines. So cell viability dropped significantly after receiving the irradiation of NIR laser, radiotherapy, and even more with the combinational treatment. So this kind of theranostic, it should have a laser or radiotherapy. So the light source should be there to activate the activate the drug, or it initiate to release the drug from the complex. Okay, so the clear indication that gold, platinum, nano dendrites would work as a photothermal agent and radiosensitized. So in this, it involved two different concepts. One is photothermal, another one is a radiosensitizer. So the gold, platinum, nano dendrites also possess a significant CT imaging signal enhancement that has the potential to guide PTT or RTT for cancers. So this is maybe act as the diagnostic as well as the treatment. Once it's get diagnosed the particular cancer, the same concept will be used to cure or used to treat the particular cancer stage or cancer cells. So this is some of the theranostic agents, examples of photo responsive metallopolymer nanoparticle for the cancer theranostic. I think no need to read all the name of this metallopolymeric nanoparticles, because this already proven that it can be used for a cancer therapy. So we have lot number of theranostic agents or theranostic metallopolymeric complex or nanoparticle for a cancer treatment, diagnostic and treatment. We have additive therapies as well. As I said, additive means additional effect. We can call it as a synergistic effect. So the synergistic combination of polymer, coated copper and selenium nanoparticles and X-ray induced radiotherapy for improved nursing care, human prostate cancer cell line. The polymer used for this additional therapy is a polyethylene glycol 5000 and polylactic acid PLA 2000. Two metals were used to prepare the polymer coated copper selenium nanoparticles. From this concept, what they observed, what they reported here is efficiently kill the prostate cancer cells. This is actually an example for LNCAP and the DU145 is a cancer cell for a prostate cancer cell line. So that effectively kill the prostate cancer cell. So apoptosis assay induced PEG PLA copper selenium nanoparticle plus X-ray was higher than that of a plain PEG PLA copper selenium nanoparticle alone. So if you have another additional agents like X-ray, photo sensitizer or light, that may be produce a more effect. So from the previous published literature, we understand that so with the polymer nanoparticle, metallic nanoparticle polymers alone also cannot do the effective cancer therapy. So we can have some additional agents like X-ray or photosensitive or some additional agents to enhance the efficacy. So the picture says that this is actually an untreated cell line. If we added the polymer loaded metallic nanoparticles, so still we can see here some of the cells, live cells. Maybe you can see the red color cells is a dead cell. The green is a live cell. So if you use the X-ray along with the complex, then we can see only few live cell in the third picture. So this is a different how they can measure about the apoptosis assay. Then the use of bioimaging, some metal polymers and its application. So mostly we will use they will use the polyethylene glycate dendrimers entrapped gold nanospheres that's used to compute the tomography imaging, demonstrate the alteration in density higher than the omnipate. So iodine based contrast agent. So it's used for a CT scanning, CT imaging. Next one is a glutathione responsive hyaluronic acid coated iron oxide nanoparticles. This is used in a magnetic resonance imaging, MRI scan. they will use glutathione responsive hyaluronic acid coated iron oxide nanoparticles. Then the third one is a polyvinyl pyrrolidine modified zinc oxide quantum dots. So quantum dots size is higher than the normal nanoparticles. It's used in the photoluminescence imaging. Then again the polyethylene glycate dendrimers encapsulated silver sulfide quantum dots used for a NIR2 biological imaging. So the nanoparticles loaded with the polymeric metallic nanoparticles loaded with polymers used for a bioimaging, mainly in CT, MRI, or photoluminescence imagings. So some of the characterization study, okay. So far we studied about the different application. Synthesis also we covered. So after synthesized, then how we will characterize whether you prepared a um, metallic loaded nanoparticle in a polymeric matrix. So, in a various techniques are typically employed for a nanoparticles characterization, which includes the transmission electron microscopy, atomic force microscopy, or scanning tunneling microscopy imaging to determine the size and surface morphology. Okay, after prepared the or after synthesized the metallic polymeric nanoparticles, so we need to characterize its size and surface, whether what is the size of prepared nanoparticles, then what is the morphology. So that can be confirmed by using the TEM or SEM or AFM analysis. In addition, the electron spin resonance spectroscopy or nuclear magnetic resonance spectroscopy, X-ray absorption or free structure, X-ray energy dispersive spectroscopy, and energy dispersive X-ray spectroscopy are commonly used to determine the structure and composition. The first one, SEM and TEM, is for a surface morphology and size. This one, the NMR, ESR, EDX, EDS, and EXAFSL and all is used to determine the structure and the composition of a metallic polymeric nanoparticles. Some other methods like UV, infrared spectroscopy provided the valuable information about the plasmonic surface as well as the chemical group and bonding on the particles. So we need to go with a sophisticated as well as the common instrument to characterize the metallic polymeric nanoparticles. So some advanced instruments also is available like gel permeation chromatography and static light scattering detector is used to determine the average molecular weight of a polymeric nanoparticles. So these are all the characterization tools or characterization instrument to confirm the size, structure, composition, and the molecular weight of your prepared metallic polymeric loaded nanoparticles. So some of the drugs, some of the polymeric nanoparticles is available in the market. The Bayer Healthcare Pharmaceutical, they introduced Fridex as an iron oxide nanoparticle associated with the Dextron for MRI scanning. Was introduced in the market, then later they were discontinued due to the lack of clinical users. So they were already withdrawn from the market. Then the another one is Resovist, is actually from same Bayer Healthcare Pharmaceuticals. It's iron oxide nanoparticles associated with the carboxydextron for MRI scanning. That also was discontinued, that overtaken by the successful market introduction of the Primovest. So they have introduced the higher version of polymeric nanoparticles. So they removed the previous one from the market. So these are some of the examples of clinical trials which is ongoing or which was completed already. Magnetable, nanotherms, and aerolase is from different, mostly iron oxide nanoparticles. Some of them are polyethylene glycol loaded. Some is spherical gold nanoparticles. So the main application for those are so magnetic hyperthermia, or sometimes used as a laser activated thermal ablation, and targeting some oncogenes, the cancer. The administration route also is different, intratumoral or intravenous. So they have two different types of route of administration. They mostly used to treat the cancers. So you see that clinical trial identifiers and what is the results against the. This actually published in the European Medicine Agency. Uh, so many metallic polymeric nanoparticle is already in the clinical trial. Some of them is completed level, that means clinical trial class 3 and class 4 as well. So these are all the reference. That means I collected the literature from my for my content. So maybe you can refer the literature which I mentioned on the respective slides for the further information. If you want more information on that related to that topic. So these are my nanoparticle related publication in the past few years with my students in my lab. So we already successfully formulated the zinc oxide nanoparticles uh, and also characterized against the in vitro cell line studies. So later maybe our plan is actually to incorporate maybe the polymeric material along with the zinc oxide to prove the anti-cancer activity. How come the plain zinc oxide and polymeric uh, particle have the difference? So future plan maybe I can uh, do the polymeric nanoparticle along with my prepared zinc oxide nanoparticle for the further enhancement to treat cancer therapy. So thank you for your attention. If you have any question or anything, then maybe you can contact me through this email. Thank you. Thank you so much.
The session is open for question and answer.
We have, okay. Okay, so then maybe I can,
Sir,
I
do you
have a few
want
MCQ
to-
questions.
Yeah, if you want to show the questions from your topic, you can run it
Okay.
and
So,
we can ask the audience.
Yeah. So from this, the lecture, I have posted here a few questions. Maybe you can answer for that. I don't know how I'm going to evaluate these answers, but it's okay. So the first question is, which of the following properties limit the use of metals in the biological application? What would be the answer for this question? So we have options here. So unique optical, electrical, and magnetic property. So due to generate the radical oxygen species, it may be a toxic one or off-targeted interaction. Which of the
Chetna Dhone has given the answer toxic. There are answers in the chat box.
okay, 1 minute.
Pooja Swami is giving C and D.
I cannot able to see the. Then
Sir, kindly open the chat box. Could you see the answers? Namrata
So-
Gawli is saying say, Vishal Rathod C.
See. Okay. My question is actually Which of the following which of the following properties limit the use of metal in biological application? Toxic is actually a common terminology. It's not affect much on that. This is no A also is not a correct answer. Yeah, the correct answer is D. It's
Yeah.
off-target interactions. Yeah. Yeah, the answer D is a right answer. So shall we move to the second question? Okay, if anybody have any other concern about this answer,
Yes, if anyone has any confusion or apprehension, sir can clarify.
I'm
Like
saying the
why the
answer D is
B,
right. Yeah.
and why not C and D? I think we should move to the next question, sir.
Okay, ma'am. I moved to second question. Okay, which of the followings are the advantages provided by polymers? This is a polymeric nanoparticle. Which of the followings are advantages provided by polymers? Answer: payload flexibility, non-toxic, tendency to form a big structure, and low cost. I think in my presentation slide, I gave the characteristics and advantages, disadvantages of the polymer, right? From that, you can, have an answer for this one. Which of the followings are the advantages provided by polymers? Payload flexibility, non-toxic, tendency to form a big structure, low cost. Yeah, that is the right answer. Polyload flexibility with low cost. A and D is the right answer for this question. I think Chetna,
Chetna
Done,
Done has given the answer
the
correct.
right answer.
Yeah.
Yeah. Thank you. Then I move to the next. If anybody have any doubt on this, why this A and D, it's actually a straight answer. In my presentation, I gave these two are advantages. So from that, we can just choose these two out of 4. Okay, I move to the next question now. So which of the following materials are not suitable to be used to form a metallopolymers? Any idea? Gold, polysaccharides, solid lipid nanoparticles, platinum. Which of the following materials are not suitable to be used to form a metallopolymers? If you want to prepare a metallic nanoparticle loaded with polymer or metallic polymer nanoparticle, what are the components or needed? We need metals, we need polymers, right? Okay, then what is the answer? What would be the answer for this one? Yes, the answer C, solid lipid nanoparticles. Okay, because the lipid one is not a monomer. So the other should have a monomer. So the solid lipid nanoparticle is the right answer. I think most of them give the answer C. So very good. Yeah, answer C for that. Then I move to the next question. So metallopolymer cannot be created by the surface coating of metal with the polymer layer by layer. Whether the answer is right or wrong. Metallopolymer cannot be created by surface coating of metal with polymer layer by layer. I'm looking response from others as well. Yeah. Yes, it's a correct because the layer by layer is one of the synthesis approach, right, for a polymeric metal nanoparticle. So the answer is no, because we can prepare the metallo polymers created by the surface coating of metal with the polymer layer by layer. So the answer is no. Then the next question, metallopolymer can only be used as a nanocarrier or therapeutic agent. What is your answer for this? You can use either carrier or therapeutic agent, right? Is there any other use? What is the answer for? Yeah, my question is or nanocarrier or therapeutic agent. Yeah, the answer no is the correct answer because we will use this metallopolymeric nanoparticle for both diagnosis and therapeutic agent. They can also used for diagnosis. Yes, I didn't ask only for diagnosis. So if I ask separately is only for diagnosis is yes, or is only for therapeutic is yes. If I ask both this or that, then maybe the answer is no. Am I right? Okay, should we move to the next one? Okay, which of the following method is used to determine the average molecular weight of polymeric nanoparticles? The answers options are X-ray energy dispersive spectroscopy, transmission electron microscopy, gel permeation chromatography in combination with static light scattering, energy dispersive X-ray spectroscopy. What would be the answer for this one? Which of the following method is used to determine the average molecular weight? Yeah, answer C is the right because very good. Answer C is right because the option A and D is just for a composition, a molecular composition, not a molecular weight. That the X-ray energy dispersive spectroscopy or energy dispersive X-ray spectroscopy is for molecular composition. Transmission electron microscopy for size and morphology. So the right answer is a gel permeation chromatography in combination with the static light scattering. So thank you for I think all of them are mentioned C here. So it's a right answer for this question. Ma'am, actually that's all from my side.
Okay, sir. If anyone wants some explanation, want to comment on the session or any questions, you are free to ask. This session is open for questions, and it's our fortune to have with us an illustrious researcher, Dr. Janki Raman, and he has joined right away from uh, Malaysia. So you have a great opportunity. So I think you must ask or seek explanation wherever you need.
Yes, so this is the option to interact with me if you have any doubt related this topic or general the research thingy, then maybe you can free feel to ask me. Maybe I can able to
Hello,
such issues
sir.
or me. Yeah.
Hello. Yes, sir. My question is, what are the key challenges in working or during processing of metalloparticles?
The key challenges, we can say that firstly, the toxicity of a metal. So we should choose the appropriate metal which is having the low toxicity to the normal cells. So instead of looking for the cancer, targeting, we need to first save our normal organs or normal cells. So we need to choose the metals which is more, you know, you don't have much toxicity to the normal cell at the same time, which is having the higher targeting capacity to the cancer environment. So these two things you need to these two things is important while we choosing the metals or the polymeric nanoparticle preparation for metallopolymeric nanoparticle preparation.
Okay, thank you so much, sir.
Welcome.
Thank you very much, sir, for your extensive coverage on challenges in targeting multi-drug resistant cancer and the extensive coverage on how the metals are selected for preparation of nanoparticles and how there are big achievements from the quotations that you cited against the multi-resistant cancers, and there are many drugs, the complexes, they have cleared the clinical trials, and some are in pipeline. So thank you very much, sir, for being with us today and enriching us, giving your time. So it was indeed a pleasure for all of us. And shortly we'll begin with the valedictory function, and I request you, Dr. Ashok Janakiraman, to stay with us for a short while. Our principal, Dr. R.O. Ganjiwale, will give the concluding remarks. So may I request Dr. R.O. Ganjiwale, sir, to give the concluding remarks.
So, good evening, one and all. Again, on behalf of the hyper, I again welcome back to this online platform. On behalf of the hyper, I heartily welcome all of you for this valedictory function. See, from the last 2 days, we are conducting this online national conference on the theme, that is nanotechnology in the healthcare. And this program was organized by the IQAC department and the pharmaceutical department of the hyper in collaboration with the APTI Maharashtra Trade Branch. And as the principal of the institute, I would like to extend my heartfelt gratitude to all the guest speakers for their benevolent support and accepting the invitation for today's conference. So day one, the keynote speaker was Dr. Jeenat Iqbal. She was from the Jamia Hamdard, New Delhi, and she spoken on the topic transforming the woman reproductive health, nano formulations and intravaginal drug delivery system in addressing the vaginal application and overcoming biofilm challenges. Under this topic, he covered the various aspects, like he covered what was the outline of the problem, woman reproductive health, even the vaginal applications, intravaginal drug delivery system, combating the biofilm, journey from the conventional IVDDS to nano-appended formulation. She also covered the vaginitis, a staggering burden, and the different types of the vaginitis, and all relevant information she shared with the participants. On the day one, second plenary lecture was organized by Dr. Sanjog Jain, Department of Pharmaceutics, Naipur, Mohali, Punjab. He talked on the topic design and development of nanotechnology-based drug delivery strategies for the advanced healthcare. And in that, he covered the various aspects of the topic. Under that, he talked about the new drug discovery and the development, how this nanotechnology can minimize untoward toxic and the side effects of the normal cells or the tissue. How this nanotechnologically formulated formulations can cross the anatomical barriers like a blood-brain barrier. Apart from that, he also covered all the recent approaches in the respect of the title that we decided. Then on second day, means today, the two plenary lectures are organized. One from Dr. Neena Sharma. Actually, we are very fortunate that we got a platform opportunity to interact with the Dr. Neena Sharma. She is a scientist of the national and international recognition. And unexpectedly, we got a chance to interact with her. And she is the founder member and the managing director of the Shamisha. This is one of the organization. And under her talk, she talked on this nanotechnology for the pharmaceutical development of nutrition, formulation, and opportunities and the challenges. Under her lecture, she covered the basic and knowledge regarding the nutraceutical and whatever the applied things are there, applied knowledge she shared with her, because practically she is working with the various pharma organizations. She is associated with the various pharmaceutical industries who are manufacturing these nutraceutical products. And practically, whatever the thing that is presently going on, she shared all those things. I'm very much thankful to her also. And now, just now, we have heard the lecture of the renowned scientist, Dr. Ashok Kumar Janki Raman. He is from the pharmaceutical department of the UCSI University, Malaysia. Just now, Dr. Janki Raman spoke on the metallopolymer nanoparticle for the carrier therapy. So in that, sir, I am very much thankful to you for covering this particular topic up to suitable depth and the length. So under that, you talk about the prevalence of the cancer treatment options, even the limitations of the current treatment of the cancer, choice of the nanoparticle, and newer strategies for the metallopolymer. What are the advantages, disadvantages of this metallopolymer? Apart from that, you also talk on how to select, how to choose the metals for the formulation, what are different types of the polymer, and the synthetic approaches for the polymer synthesis. So again, I am very much thankful to Dr. Ashok Kumar Janki sir for sharing your very valuable information, scientific information with the participants of the conference. Once again, I would like to thank all my participants for their enthusiastic involvement, participation, for maintaining the patience during the conference, and nobody left out during in the midway of the conference. They supported us to maintain the discipline and the decorum of the conference. I am also thankful to all my faculty members of coordination committee for their well-coordinated efforts throughout the conference. Especially, I would like to take the name of my coordination committee, Dr. Shagupta Khan, Dr. DJ Singhvi, Mr. Abhiram Deshmukh, SP Devani, Budrani, then Hadke Madam, and everyone who are directly or indirectly associated with the organization of this national conference. I am also thankful to my managements of the VYWS for their continuous motivation and the encouragement to conduct such type of the national conference. And I am also thankful to all my other faculty members also who directly and indirectly supported for the successful organization of this national conference. Thank you. Thank you very much.
May I request Mr. Abhiram Deshmukh to seek comments from the participants and then propose the vote of thanks.
Thanks a lot, madam. Let us come to the views from some of the participants or delegates of this conference. May I call upon Dr. Nishan Bobde, Associate Professor, Vidya Bharati College of Pharmacy, Amravati. Dr. Nishan Bobde, please, if you can share your views regarding.
Thank you, sir. Good afternoon to all. I am feeling very privileged to express my view as a participant on this occasion. I am thankful to Dr. R.O. Ganjewale, sir, Principal, Hyper Varda, Coordinator and organizing team members of APTI sponsor online 2 days national conference on nanotechnology in healthcare. All the eminent speakers of the conference were the very knowledgeable and versatile personality in pharmaceutical sector. They offer valuable information and provided a bird eye view on the current scenario in nanotechnology in healthcare. Professor Dr. Zenith Iqbal Madam expressed her knowledge on women's health and intravaginal drug delivery system very thoroughly. Professor Dr. Sayong Jain sir presented nanotechnology research challenges in formulation and characterization very nicely. Professor Dr. Neena Sharma Madam explained very properly on topic nutrition formulation. Professor Dr. Ashok Kumar Jankiram sir delivered the brainstorming content for the cancer therapy and gave proper direction for nanotechnology development. All the sessions of the conference were extremely informative for the researcher in this sector. Overall conference was well planned, organized, and coordinated in systematic manner on this platform. I would like to participate in such conference in future. I am once again thankful to Professor Dr. Shagupta Khan Madam for providing opportunity to speak on this occasion. Thank you, Madam.
Thanks a lot, Nishan sir. May I call upon Dr. Mithali Bodhankar Madam from Guru Nanak College of Pharmacy, Nagpur. Dr. Mithali Bodhankar Madam, Professor, Guru Nanak College of Pharmacy, Nagpur.
Thank you, Abhiram sir.
Thank you.
Thank you very much. It was indeed a pleasure to be a part of this APTI-sponsored online national conference as a participant on the topic nanotechnology in healthcare, organized by Institute of Pharmaceutical Education and Research, Wardha. I will congratulate Dr. Ganjiwale and Dr. Shagufta Khan Madam, Dr. Dilesh Singhvi, and Abhiram Deshmukh sir for organizing such a nice conference. Because the seminars and conferences, they give a lot of insights and knowledge to the upcoming pharmacists who are working in particularly research area. The conference was organized nicely, and the keynote address was efficiently delivered by Dr. Zenith Iqbal from Jamia Hamdard, New Delhi. She talked on women's health particularly, and really we see that women's health matters and as it affects the society. And she talked on transforming women reproductive health, the nano formulations, the intravaginal drug delivery system, and the biofilm challenges, and how they can be overcome. She talked about the antifungal based lipid nanoformulations, the nano emulsions for biofilm associated candidal vulvovaginitis, which is the most important area in research nowadays, and particularly which may lead to some cancerous growths also because of the viral and bacterial load. And that is why the women's health must not be neglected, and she has delivered it very nicely. The second session was conducted by Dr. Sanjoy Jain, and on the design and development of nanotechnology based drug delivery strategies for advanced healthcare. The amount of the drug which is needed to be taken in the nano formulation, he just calculated and gave the calculations, and that was a very important part of his delivery. The nanocolloidal drug delivery systems, the pegylated nanoparticles, receptor mediated endocytosis, also he explained very nicely. He gave the insights on ligand mediated active drug targeting, the folate PEG HSA nanoparticles, and with the examples of his patents, and the parental versus mucosal immunization, and he gave the various insights for the research to the students. Then on the second day, the first lecture was conducted by Dr. Neena Sharma on nanoformulations in nutraceuticals. Um, uh, Basically, the health, when we talk of the health, the nutraceuticals or the nutrition cannot be neglected. Prevention is always better than cure. And healthy eating pyramid, she has started her talk with this concept, the probiotics and the prebiotics, and then on oncology supporting therapy, and the bioactive compounds in nutraceuticals. The most important part in her speech was the glutathione Parkinson support therapy and the innovation in packaging. And it gave the various insights for the entrepreneurship also, because this is the area which is emerging in this direction. The last lecture was by Dr. Ashok Kumar Janaki Raman, UCSI University, Malaysia. It was a wonderful talk on metallopolymer nanoparticle for cancer therapy. And it was enriching knowledge about the drug delivery of nanoparticles to treat the cancer. Polymer based, inorganic metal type, lipid based, carbon based nanoparticles, he has explained very well. The nanotoxicity issue, yeah, he has very well explained, and the strategies which are to be followed: surface coating, entrapment within polymeric matrices, and the utilization of the polymeric capsules. The he also he has also thrown light on biosensors as diagnostic tools, and theranostics and bioimaging. It was a wonderful talk, and giving insights to many of the young budding researchers. And I'm very much thankful to IPER Vardha for organizing such a nice conference. Particularly, such conferences are to be arranged to give the insights for the further research to the young upcoming pharmacists who are working in the area of research. So, thank you very much. Thanks for giving me the opportunity to say a few words. Thank you.
Thanks a lot, madam, for your kind remarks. May I call upon now Ms. Kajal Chaudhary, Assistant Professor from PR Pote College of Pharmacy, Amravati. Ms. Kajal Chaudhary, Assistant Professor, PR Pote College of Pharmacy, Amravati.
Thank you, Abhiram sir, for giving me a chance to be a part of conference. We all know That conference attendance is the life wire of all academics, and they connect many academics together to share their view about a particular theme. In our today's conference, that is nanotechnology in healthcare, the guest present here is Zeenat ma'am, Sayog sir, Ashok Kumar sir, and Sharma ma'am. They shared their gold words about nanotechnology, and they shared all research related work in nanotechnology. And this will be a very informative session, sir. Thank you all the guests for such a valuable session, and my special thanks to uh, committee members of IPER and the family members of IPER for organizing such a great national conference. Thank you, sir. Okay, thank you, sir.
Thank you, Kajal. Thank you very much. Last, may I call upon Miss Apurva Tiwari, research scholar, Dada Saheb Balpande College of Pharmacy, Nagpur. Miss Apurva Tiwari, research scholar, Dada Saheb Balpande College of Pharmacy, Nagpur.
Thank you, sir. I want to appreciate and congratulate Dr. Gajjiwale, sir, Principal, Dr. Shagupta, ma'am, and the team of IPO for organizing such informative session. The session was well organized and coordinated by all the coordinators. The session taken by Dr. Zeenat, ma'am, on intravaginal drug delivery system, and session by Dr. Sanyog, sir, Dr. Sharma, ma'am, Dr. Ashok Kumar, sir, on types of polymer in nanotechnology and drug delivery system gave different insight about the research. All the professionals delivered outstanding fruitful sessions, and I would like to join more such valuable sessions in future also. At the end, I want to thank Mr. Abhiram Deshmukh sir for this opportunity. Thank you.
Thanks a lot, Apurva. Now, may I call upon Miss Neha Gallani, Assistant Professor, Institute of Pharmacy, Badnera. Miss Neha Gallani, Assistant Professor, Institute of Pharmacy, Badnera.
Good afternoon, everyone. This is Ms. Neha Galani, Assistant Professor at Institute of Pharmacy and Research, Badnera. I am very fortunate and privileged to attend this 2 days APTI-sponsored national conference on nanotechnology in healthcare, which was held from 6th to 7th October. All the sessions were very well planned and conducted smoothly, and a big thanks to the organizing team for the great initiative for bringing all the faculties together on a common ground so that we can interact and enrich ourselves. We interacted with the eminent personalities from the pharmaceutical field, Professor Dr. Zeenat Iqbal ma'am, who explained the role of nanoformulations and intravaginal drug delivery systems in addressing vaginal affections. Professor Sayog Jain from NIPER, who explained the various nanotechnology-based strategies and nano carriers for advanced healthcare. Dr. Ashok Kumar from UCSI University, Malaysia, who explained the red light responsive metallopolymer nanocarriers, strategies in creating the metallopolymer nanocarriers, choices of nanoparticles, and theranostics and additive therapies in cancer. Also, Dr. Neena Sharma explained the nutrition of patients, nanotechnology-based pharmaceutical development for nutraceuticals and opportunities and challenges. Thank you for this great opportunity. It was a much required session, and we hope to attend such more sessions in future. Thank you so much.
Thank you, everyone. If anybody from delegates or any other participant want to share their comment, they can share their views regarding the conference. Okay. So good evening, everyone. We are towards the end of 2 days online national conference now. I am pleased to be standing here on behalf of Institute of Pharmaceutical Education and Research, Wardha, to convey my gratitude. First of all, I extend my most sincere thanks to the Almighty God for making today's event resoundful success. With His blessing and grace, we are able to make this event what it was. I would like to start by thanking Dr. Nitin Dhande sir, President of Vidarbha Youth Welfare Society, Amravati, and all the respected members of the management, who have always been supportive of our initiatives and encourage us to strive for excellence. I need to mention my deepest sense of appreciation towards Dr. Siri Jain sir, Vice President of APTI Maharashtra State, for his kind support and cooperation. A particular mention goes to Dr. R.O. Ganjiwale sir, esteemed Principal of Institute of Pharmaceutical Education and Research, Wardha, for being the catalyst that stimulated us to do our best and standing as pillar of strength. As we all know that saying thank you is a respectful way to show appreciation towards a person, but these words would be less for our guest speakers of the conference. Professor Dr. Zeenat Iqbal, Jamia Hamdard, New Delhi, Professor Dr. Sanyog Jain sir, Naipur Mohali, Dr. Neena Sharma, Managing Director, Shamisha Resources Management Company, Mumbai, Dr. Ashok Kumar Janaki Raman, UCSI University, Malaysia, who deserve special recognition for accepting our invitation, being here with us, and throwing lights on various aspects related to nanotechnology in healthcare. I would also like to extend my gratitude to Dr. Shagufta Khan Madam, coordinator of this conference and professor and head of the pharmaceutics department of IPER, whose unwavering support has been instrumental in growth and development of Department of Pharmaceutics of the Institute. A special thank goes to Dr. Dilesh Singhvi sir, coordinator of Internal Quality Assurance Cell of IPER, who took the responsibility of this conference to be a success. It isn't something that happens overnight. Weeks ago, the wheel began to turn. It requires planning and a bird's eye for detail. We have been lucky enough to be backed by the support of team of proactive and devoted members of our institution's personnel who are experts in their field. I would be falling short of my responsibility if I did not express my gratitude to my co-workers. Dr. Jayashri Wankhede Madam, Mr. Sunil Devani sir, Mr. Ashish Budrani sir, Ms. Priyanka Savarkar Madam, and Mr. Samir Asole sir. Let us give applause and heartfelt appreciation to all teaching and non-teaching staff of IPER who contributed to make the conference a success. Around 350 delegates participated from different institutes of Maharashtra and also from other states upon invitation. This made the conference a grand success as it was able to attract the attention of all the curious and motivated faculties, research scholars, UG and PG students who are willing to learn and explore. Learning is always a good opportunity to feed our mind as it helps us to grow. Through such informative conversations, we get a chance to boost our knowledge with a unique opportunity to dive into a field of subject that might be unfamiliar to us. So I would like to take my time in thanking our dear audience who took their time to be a part of us. Finally, I would like to thank each and every one of you for your support and participation in making this event a grand success. Your contributions have not gone unnoticed, and I am grateful for all that you have done. In conclusion, I would like to say that this event would not have been possible without the support and hard work of each and every one of you. On behalf of Institute of Pharmaceutical Education and Research, Wardha, I would like to express my sincere thanks to all of you. Thank you very much. Finally, I would like to hand over the session to Dr. Shagufta Khan Madam, coordinator of this conference, professor and head of the pharmaceutics department of IPER, for her final conclusive remarks. Dr. Shagufta Khan Madam.
Thank you, Abhiram. At the end, I would like to say that a prayer like thank you cannot be seen or touched. It has to come from the heart. And from the bottom of my heart, I would like to mention that the conference would not have been successful without the response by the participants. My heartfelt thanks to all My heartfelt thanks to all the honorable delegates who blessed us with their presence. Your kind words means a lot to us and acts as an impetus for organizing this kind of conferences in the future. I am equally thankful to all the speakers to make this conference a successful one. I express my deep sense of appreciation to the management of the Institute for providing us the opportunity and motivating us for conducting conferences like this on the most advanced technologies having capability to change the dynamics of human lives. My sincere thanks to our beloved Principal Sir Dr. R.O. Ganjiwale for his constant guidance and motivation that radiated as the source of energy within us. I am indebted to my committee members, Dr. Dilesh Singhvi, Mr. Sunil Devani, Mr. Abhiram Deshmukh, Dr. Jayashree Hadke, Ms. Priyanka Savarkar, Mr. Ashish Budrani, and Mr. Samir Asole. Their enthusiasm and dedication towards making this conference successful was commendable. Many thanks from my bottom of heart to the supporting staff for their incredible support. I feel proud and thank you all for making this conference a successful one. With your appreciation, certainly we can organize a lot more events like this in future. Thank you very much, all of you. There is an announcement by Mr. Abhiram Deshmukh. So may I request him for the same?
All the participants of the conference are further requested to fill 2 Google Forms, links for which have been posted in the chat box. One feedback link is for the feedback of this session, and another link is providing for providing your details to be included in the participation certificates. You are kindly requested to be careful while filling your details, such as prepositions, candidate name, institute name, designation, etc., as this data will be used for including in the certificates. You will get your certificates within 1 week from now, so don't forget to fill both the links which are posted on the group. So in chat box there are 2 links. Kindly fill both the links. One is for the feedback of session and another is for data related to the certificates of participation. Thank you, everyone. I further request all of you to please switch on their video cameras so that one common group photo can be clicked or screenshot can be taken. I request all of you.
Ashok Janki Raman sir also. Sir, are you hearing us? Dr. Janki Raman?
Yes, yes, I am here.
Yes. Please switch on your video, sir.
Yeah.
Everyone, all other participants are also requested to switch on their cameras. Good. Many of you still have not switched on your cameras. Please switch on. पहले 2 स्क्रीन में सर। मैडम कितने आ रहे? Okay, thank you, everyone. Don't forget to fill those two feedback links. One is for feedback of the session, final session, and one will be for providing the data related to participation certificate. Your participation certificate will be shared to you within a week.